Chapter 15, the second line of defense. The learning objectives for this topic, and there's quite a few of them, so hold on. Compare and contrast the body's first and second lines of defense against disease. Discuss the components of blood and their functions in the body's defense. Explain how macrophages are named. Name and describe the 6 stages of phagocytosis. Describe the role of eosinophils, natural killer cells, abbreviated NK, and neutrophils in non-phagocytic killing of microorganisms and parasitic helminths. Define toll-like receptors and describe their action in relation to pathogen-associated molecular patterns. Describe the location and function of NOD proteins. Explain the roles of interferons in innate immunity. Describe the complement system, including its 3 activation pathways. Discuss the process and benefits of inflammation. Explain the benefits of fever in fighting infection. In this topic, we're going to discuss the defense components of blood, phagocytosis, non-phagocytic killing, non-specific chemical defenses against pathogens, inflammation, and fever. First, let's talk about blood. Blood comes in 2 parts, the liquid portion, known as plasma, and the solid portion, which comes in the form of cells and cell fragments. Now we're going to talk about the chemical defense components of plasma later on in this topic. For now, let's go into the cells. All blood cells come from hemopoietic blood cells, or blood stem cells in the bone marrow. From there, they split into 3 different lineages. One of them ends up becoming your red blood cells. Another one ends up becoming platelets and granulocytes. They are called this because when you stain them, you can see little granules all over in the cytoplasm. Now these granules are full of enzymes that are important in the functioning of these types of cells: the basophil, the neutrophil, and the eosinophil. Now basophils stain with basic stains, usually crystal violet or methylene blue. Eosinophils stain with the acidic stain, eosin, and neutrophils stain with both of them, which gives you an indication of what the pH of the granules are. Also, from this stem line, we get a granulocyte called the monocyte. We also get lymphocytes. One type of lymphocyte, the natural killer lymphocyte, is involved in innate immunity. The other 2 types of lymphocytes, B cells and T cells, are involved in adaptive immunity, and we're going to talk about those in the next chapter. All of these cells pretty much stay in the blood vessels unless we're involved in inflammation. Now we're going to talk about inflammation more later, but as kind of a preview, during inflammation, the cells of the capillaries contract, pull away from each other, leaving spaces for the cells to leave and move into the tissue where there may be a possible infection. We call this process diapedesis, or immigration. And this is the process of them squeezing in between the epithelial cells of capillaries. Now monocytes, generally speaking, wander about the body in capillaries, but eventually they will leave the capillaries, move into the tissues, and mature into macrophages. Now we're going to talk about the functions of basophils, neutrophils, eosinophils, monocytes, and natural killer cells during the course of this topic. First, let's talk about phagocytosis. Of the cells that we talked about in the previous slide, macrophages are the ones that do the bulk of phagocytosis within the body. The others, basophils, neutrophils, and eosinophils, do a little bit of phagocytosis. It's not their main job. But let's talk about what phagocytosis is and the process of it. Phagocytosis is where an animal cell, a cell without a cell wall, engulfs large objects. It's Latin for cell eating. Now, the first thing that happens in phagocytosis is chemotaxis of the phagocyte to the microbes. So, chemotaxis is movement of a cell toward a chemical. In phagocytosis, within the human body, the chemicals that it moves toward are called chemotactic factors. Chemotactic factors are chemicals that are released by injured cells. They're released by other cells in the immune system, and they are chemicals that are also released by the microbes. In any case, the phagocyte moves along the chemotactic trail, sort of like a bloodhound, and come in contact with the microbe. We call this adherence or attachment. In other words, before it can swallow it in, it's got to get a hold of it somehow. Now, this process is made easier or accelerated by opsins, and we call this opsonization. Now, opsonization is Latin for to make tasty. In other words, if something has opsins on it, the phagocytes eat them faster, or phagocytize them faster, as if they were tasty. There are several molecules that are opsins that we're going to be talking about in detail, but to give you a preview, complement is an opsin, antibodies are opsins, and all of them, once they're stuck to the microbe, make it easier for the phagocyte to grab a hold. Now, once the microbe is adhered to, it is ingested by the process of phagocytosis. The cell membrane wraps around them, pinches off, and we form a phagosome. Meanwhile, being pinched off from the Golgi body are lysosomes. We talked about lysosomes before. They are full of hydrolytic enzymes, and they have about a pH of 5.5. The hydrolytic enzymes work best at this pH, and we keep them wrapped in lysosomes so that they don't escape into the phagocyte and eat up the phagocyte. So once the lysosome fuses with the phagosome, we call this a phagolysosome, and the hydrolytic enzymes go to work, breaking down the microbe. Now, there are other things that are also in the lysosome. There are oxygen radicals, like superoxide radicals and hydrogen peroxide. Now, remember, we talked about those in the chapter on oxygen requirements of microbes when we're growing them. So some microbes are going to be very sensitive to these, are going to be killed by these. They also release nitric oxide, which also kills the microbes and is also a chemotactic factor. And then also they have inside of them hypochlorite, which is the active ingredient in bleach. So we've got all of these antimicrobials that work to break down the microbes within the phagolysosome. Some of the breakdown products, amino acids, carbohydrates, lipids, a little bit of nucleic acids, can be used as food, as sources of energy for the phagocyte. Whatever's left over is exocytosed by the phagocyte. Spits them out. Later on, we're going to talk about, in the next chapter, what we do with some of these other than spitting them out. Phagocytes are all antigen-presenting cells, and the leftovers in these residual bodies are antigens that need to be presented to the adaptive immune system. Now that we've talked about the main job of macrophages, let's talk about the jobs of the other cells of the innate immune system, most of which falls under the category of non-phagocytic killing. Now, I've tossed in basophils. Basophils are not involved in non-phagocytic killing, but this is a good opportunity to talk about what they do. Basophils, as you may remember, stain with basic stains, and their main function is the release of chemotactic factors. In other words, they're aware of a pathogen in an area, they release chemicals that bring in phagocytic cells, and some of these other cells that function in non-phagocytic killing. Now, one of the main components of basophils and their cousins, the mast cells, is histamine. You've probably heard of histamine, especially if you have allergies. When histamine is released due to allergens, which are antigens or proteins that stimulate the immune system but are not actually pathogens, then we get an allergic response, and we're not happy with it, so we take antihistamines. Let's move on to eosinophils. Eosinophils stain with acidic stains, such as eosin, and when we take a blood sample and we find larger than normal amounts of eosinophils, that indicates that we have a worm infection or a helminthic infection. Eosinophils cluster around multicellular organisms or parasites that are too big to phagocyze and basically drop antimicrobial bombs on them. Eats away at the skin, the cell membranes, etc., and kill the invaders. That's the main function of eosinophils. They also phagocytize, but they'd really prefer to drop these antimicrobial bombs. Neutrophils, as you may remember, stain with both acidic and basic stains. Their granules have a neutral component. Now we're going to talk about neutrophils again later. I love repetition, it helps you remember. But neutrophils are the first responders, generally speaking, to the site of an injury or an infection. And they do phagocytize. They phagocytize more than the basophils or the eosinophils, but not as much as the macrophages. And like the macrophages, they produce oxygen radicals, hypochlorite, and nitric oxide, but instead of producing them in phagosomes, which they're capable of, they have enzymes on their cell membranes that produce them outside. So if we have bacteria out here, they're being exposed to oxygen radicals, the active ingredient bleach, and we're also releasing nitric oxide, which kills them and acts as a chemotactic factor, bringing more help to the site. Now, one thing to mention is these things not only affect pathogens, but any cells that happen to be nearby. If you have the site of an injury, say a cut, a lot of those cells are going to be dead anyway, and they have to be cleared by phagocytes to get them out of the way so that we can build new tissue. But during chronic inflammation, that's part of what causes diseases that are related to chronic inflammation. So it's good not to have these guys working long term. Just want to have them work short term. Also, if things get really bad, they will produce what's called a NET, or a neutrophil extracellular trap. What they will do is they will burst their nucleus, and they will mix their DNA with the granules, toxic granules, and then they will explode, throwing out the DNA onto the surrounding cells with these toxic granules. In other words, they're throwing a net over everything. Results in a broader range of killing, and actually it's kind of cool. Now over here we have our A granulocytes. Here we have our macrophage, which we've already talked about, and here we have our natural killer lymphocytes. Lymphocytes do not phagocytize, but they are engaged in non-phagocytic killing. Now, they do this in the same way that cytotoxic T cells do, and we're going to talk about that process in more detail in the next chapter. But what I want you to remember is they go around checking your cells for anything that means something's not right. In other words, cancer cells that aren't producing the right markers, or virally infected cells that aren't producing the right markers on their surfaces. And they say, oh, sorry, we're going to have to kill you. And they drop little bombs on them, kind of like the eosinophils do, but these bombs tell the cells to self-destruct in a process that is called apoptosis. So natural killer cells help suppress tumor development and helps clear out virally infected cells.